Okay, let's proceed to the topics no under dietary lipids. So I'd like to emphasize particularly this one, no, lipoprotein metabolism, because I know you have mastered the digestion and absorption of fats already, but I think kinahanglan pag further emphasis no ang unsay mahitabo after absorption, and that's transport of lipids in the body. So we have three types of lipid transport through lipoprotein metabolism. We have exogenous, endogenous, and reverse cholesterol transport. So each of these types is being coordinated by a particular set of lipoproteins. So for exogenous, it's being coordinated by chylomicrons and chylomicron remnants. For endogenous lipid transport, it's coordinated by very low density lipoprotein, intermediate density lipoprotein, and low density lipoprotein. And we also have reverse cholesterol transport, which is um, coordinated solely by high density lipoprotein. So first, we have to review first no, the structure of a lipoprotein. So just like what micelles are, 'di ba ang micelles kay ang naa sa gawas maunang um, hydrophilic, maunang mag-interact sa aqueous environment, maunang water soluble. Pero kanang naa sa sulod, maunang hydrophobic, maunang ginasulod siya kay hydrophobic man siya, di man siya mag-interact with water. So that's why these hydrophobic compounds are being internalized. So lipoprotein, just like micelles, is a kind of amphipathic structure. Okay, when you say amphipathic, both na ay hydrophobic and hydrophilic structures. When you say non-polar, it's also associated with hydrophobic compounds, while polar compounds are those associated with hydrophilic or water-soluble compounds. Okay. So of course naa sa sulod kaning cholesterol ester, cholesterol nga esterified with fatty acid. Of course naa ang fat or triglyceride molecule sa sulod. Okay? So the core is mainly non-polar lipids. Tapos naa dayon another layer um after this core non-polar lipids, and this is now your phospholipids. Okay, nga ang head mauning um, hydrophilic and ang tail hydrophobic. Okay, and then kaning mga dagko nga spherical structures nga at either trans or sa surface gyud. So when you say trans proteins, no, it crosses um, the exterior or the membrane. Okay, so ang tawag is integral. When you say integral, it crosses no the layer of phospholipid. Okay, such as kani. Whereas kaning naara sa surface ga float maunang peripheral apoprotein. So usually kaning integral apoprotein na apod siya hydrophobic structure that can interact with the hydrophobic interior, and usually kani siya mga hydrophobic amino acids pod ang naa diri sa sulod. Then ang naa sa gawas, since ang gawas kay aqueous, so mao pod na mga hydrophilic nga amino acids. Okay? So these are proteins, guys. So therefore, ang imong lipoprotein na ay fats, na ay mga lipids, and proteins. So that's why it's called lipoprotein, because it's a combination of lipids and proteins. So the dietary lipids that can be found in the interior part of the lipoprotein is again your free cholesterol or esterified cholesterol or cholesterol esters, and of course the fats or the triglyceride molecules. Okay? And yeah, naapod sa gawas guys ang free cholesterol, no. Okay? So this is the generalized structure of a plasma lipoprotein. And this has many types. And kani sila actually. So we have the HDL, LDL, IDL, VLDL, and chylomicron. So actually ang pinakadako no is the chylomicron because it has the most amount of fats. When you say fat, again, it refers to triglyceride molecule or triacylglycerol. So 82% of a chylomicron is composed of tags or triacylglycerol or fats, di ba? 7% ang phospholipid, 2% ang cholesterol, and 9% is proteins. So kaning mga proteins, this is made up of kaning mga apoproteins. Okay. And yeah, ang order dayon no kay decreasing order dayon. So ang pinakagamay is HDL. Pero actually sa kabug-aton, ang kabug-aton actually no is measured by the number of protein. And actually the heaviest or the most dense lipoprotein is the HDL since 50% of it is composed of proteins. Diba? So mas dense ang HD, pinaka-dense ang HDL. Although pinakagamay in size, but pinaka-dense because siya ang naipinakadaghan nga proteins. Okay? Unsay pinakadaghan og cholesterol? We can see here it's the LDL. Oh, 47% of the LDL is made up of cholesterol. Okay? So I would like you to rank these lipoproteins no, according to decreasing order of fats, decreasing order of cholesterol, decreasing order of phospholipid, and decreasing order of protein. For example, decreasing order of protein. So balihon nimo no HDL, LDL, IDL, VLDL, and then chylomicron. Okay? So inana siya. Kung decreasing order of cholesterol, then we have um, LDL, IDL, VLDL, HDL, and then chylomicron. Diba? So, inana siya. Naani siya, this would make more sense later no, as we discuss this. Kay, for example, ma-explain no, nganong ang LDL kay siya ang pinakadahan ng cholesterol. Nganong ang HDL or ngano ang chylomicron is nganong siya ang pinakadaghan og fats. And then nganong gaka-decrease man ang fats if you arrange VLDL, ADL to LDL nga order, di ba? So later ma-explain na siya. But first, let's discuss no, these proteins that are integrated in the lipoproteins. So these are called apolipoproteins or simply apoproteins. Okay, so these are either again integral or peripheral. Okay, so unsay function ani? Ang general function ni proteins nimo sa lipoproteins, nganong naa may proteins sa lipoproteins, ay ang general function is first to activate the enzyme nga needed no para ma-utilize ang components sa lipoproteins. And ikaduha is they serve as a ligand for receptors. So remember, unsa gay ng ligand maunang mo receive maunang mo bind sa mga receptors sa cells to to para ma-activate ang cellular signaling or cellular communication. Okay. So pinaka-important din no is ApoC2, Apo100, and ApoE. Okay. Kay mauna sila ang naay prominent nga mga functions. So, kaning ApoC2, siya amo activate sa extrahepatic lipoprotein lipase. So, lipoprotein lipase is an enzyme that would help release the fat that is contained in the lipoprotein and for it to be able to be hydrolyzed to monoglycerides and glycerol para magamit siya sa extrahepatic tissues. When you say extrahepatic tissues, these are the tissues that are not the liver. So in this discussion, we have the adipose tissues and then the muscle tissues or the skeletal muscle tissues. Okay. APO100 is the ligand for the LDL receptor kani kay naa ni siya relevance especially para mas masabtan nato nganong ang LDL kay ginaingon man nato nga bad cholesterol, di ba? Yeah. So mauni ang mo receive mauni siya ang kanang need ni LDL receptor para para ma-activate siya. Then si ApoE morning ligand for chylomicron remnant receptor, which we will also discuss later. So sa una sa basic nutrition we just discussed um unsay ginaingon nga good cholesterol which is your HDL, and then bad cholesterol which is the LDL cholesterol. But karon, since we are in nutritional biochemistry already, so let's discuss the in-depth details regarding these lipoproteins. First, ato sa siguro himay-himayon kaning exogenous lipoprotein metabolism. So, exogenous lipoprotein metabolism is when the body wants to transport the fats that are absorbed from the diet. So therefore mao ning gikan sa intestines. Okay? Then maugid ni ang mahitabo gyud most certainly after you ate like a meal that is very fatty or that is very high in fat. Then mao dayon ni ang mahitabo dayon. Okay? So, you know that chylomicrons are synthesized by the enterocytes, and you know nga mauna ang mahitabo during absorption. Okay? Then, para ma-absorb sa body ang digested fats and other dietary lipids, kinahanglan nga incorporate ni sila sa chylomicrons and ipagawas first through the lymphatic system and then to the portal hepatic circulation. Okay? So, naatay mga steps diri kani summary ni siya, nindot ni siya nga illustration guys, kay very summarized and mas masabtan nato kung inani. Okay, so morning step 1. So chylomicron contains ApoB48 and ApoA1. So kani nga mga proteins, they are already synthesized by the enterocytes. Okay, so kani siya ma-produce na ni sila daan nga mga proteins sa small intestines pa lang. Okay? And these are needed by the chylomicrons. Okay? So, munang mahitabo. And then, of course, i-release na yun ni small intestines ang chylomicrons. And then, let's proceed to the step 2. So, ang step 2 na to, um, ApoE and ApoC2 are transferred to the chylomicron from HDL. So kaning mga HDL, they are just floating around. Kaning mga nascent or kaning mga immature HDL nato, kaning mga precursors nato for HDL, they are just floating around the plasma. They are just floating around the bloodstream. Okay? Dili ni sila like gikan gyud siya sa liver, no. Na ang mga or ang mga precursors to HDL naara na sila sa plasma. Okay? So kani sila mauning mo donate sila og ApoE and ApoC2 sa chylomicron nato. So as you can see, ang original nga proteins nga naa sa chylomicron is ApoB48 and ApoA1. But pag humanog transfer ni HDL ang ilahang ApoE and ApoC2, i-transfer nila kay chylomicron. So upat na ang proteins nga naa sa chylomicron. So aside from the original B48 and A, naasad ang E and C as you can see here. So unsay purpose aning nganong kinahanglan i-transfer ni HDL ang ApoE and ApoC? Because ApoC has this very important function which serves as the activator of lipoprotein lipase. So, mauna iyang purpose nganong kinahanglan i-transfer ni HDL ang kaning mga apoproteins niya. Okay? So, unsay mahitabo diri sa step 3? Chylomicrons deliver the tag to tissues other than the liver, particularly adipose and muscle. So, kato, niagi na siya sa lymphatic system, then niagi na siya sa portal vein. Padulong na ni sa um, Okay, padulong na ni siya sa circulation. Okay? So kani siya dili pa ni siya actually mo dili pa ni siya moagi sa liver. Okay? Moagi pa ni siya una sa peripheral tissues. Okay? So ang nahibaw-an ba ninyo last time is ang tanang nutrients ang tanan, okay? O naay ka ng quote diha. Ang tanang nutrients kay after ma-absorb padulong na gyud first sa liver. But actually kaning fats nato, kaning dietary lipids nato, moagi sa ni sila og peripheral tissues ay ha sa liver. So mao ning exception. Okay? So that's why kaning step 3 ni agis siya sa extrahepatic tissues like adipose tissues and muscle tissues. So, nganong kinahanglan siya mo agi diri kay iyahang i-donate ang mga fats nga naa sa sulod, ihatag niya sa skeletal muscles and adipose tissues. Okay? So, what's the purpose? For example, as a source of energy, okay, or um, adipose tissues, di ba, mo ng storage na to sa fat. So, especially if daghan kaayo gikaon nga fats, then limited ramang good ang storage sa fat sa liver. So, they have to be delivered directly to the adipose tissues for storage. Because ang atong diet karon kay grabe gyud siya sa kataas sa fat, no. So it is expected nga kaning mga fats kinahanglan i-transfer to the adipose tissues for storage. And what enables that? It's actually the ApoC. Okay? Because ApoC activates the enzyme which is called lipoprotein lipase, which again hydrolyzes. First, i-release niya ang nasa sulod nga fats, then si lipoprotein lipase dayon iyahang i-break down no ang fat into monoglyceride and glycerol. Okay? So, mauna siya para magamit sa kaning mga tissues. Pero kaning 4, nganong naay 4 diri nga labeled sa glycerol? Because the muscle tissues cannot phosphorylate glycerol. So they transfer it to the serum to be picked up by the liver or kidney. So kaning glycerol gamiton ni siya for gluconeogenesis. And the only tissues in the body that can do gluconeogenesis is the liver and the kidneys. So that's why, remember, kung ma-hydrolyze ang ma-hydrolyze ang fats, di ba, na ay mo ma-produce nga glycerol, okay, ma-break up man ang glycerol from the three fatty acids. So kaning glycerol mauning i-donate niya sa mga tissues that are capable of gluconeogenesis. Okay? And glycerol is also used para kaning mga fatty acids ma-esterify, ma-combine sa iyaha para makaproduce og another fat molecule. So the one that can do that is liver, because the liver can, can esterify fatty acids to glycerol and make fats from it. Okay. The muscles cannot simply do that, so that's why the muscles must give up this glycerol para ma-process sa mga organs that are capable of processing it. Okay. So step 5, when much of the tag are transferred from the chylomicrons, they became chylomicron remnants. So 'di ba iyahaman ihatag ang mga fats sa extrahepatic tissues. So as time goes by, mugamay in size ang chylomicrons. Kaya iyahaman i-sacrifice, iyahaman ihatag ang mga fats. So mugamay siya in size. Okay? And later, mahimo siyang chylomicron remnants. So kaning chylomicron remnants na to, depleted na ni siya sa fats. Okay? Hapit or kung dili man galing completely depleted, then hapit na ma-exhaust. Okay? And this is where the role of ApoE comes into play, because ApoE serves as the ligand for the chylomicron remnant receptors nga naa sa liver. So maning mahitabo sa step 7. Okay? So, ang step 5, ang chylomicron mahimong chylomicron remnants. Ang step 6, the chylomicron remnants transfer the ApoA and ApoC back to the HDL. So para ma-recycle ang ApoC kay wa naman siya gamit kay nahatag na niya tanan nga fat sa extrahepatic tissues. So the chylomicron remnant has to give back the ApoC to the HDL. Okay? And if ang nabilin is ApoE na lang and ApoB48, then mauna ng step 7. So, due to the interaction of the ApoE with the receptor in the liver, so kaning si chylomicron remnant pwede ni siya magamit sa liver. So, usually iyahan ni i-absorb ang chylomicron remnant and i-process niya ang mga nabilin diha um, para makahimo og additional cholesterol maybe or kaning mga cholesterol nga nabilin diha iyahang i-transform into cholesterol esters or kaning mga cholesterol iyahang i-convert to bile because remember that the precursor of bile synthesis is cholesterol. Okay? So yeah, this is the exogenous lipoprotein metabolism only proceeds in 7 phases as you can see here. Okay? So yeah, I hope nakuha ninyo. Very simple naman, di ba? So, ang iyahang goal ra gyud ani is iyahang ihatag ang mga na-absorb nga fats gikan sa diet, iyahang ihatag sa mga muscles and adipose tissues. Then ang nabilin, that is the remnant, i-process dayon ni liver. Okay, so take note of the apoproteins that are needed uh, in those purposes. Okay? So si ApoC kinahanglan niya kinahanglan ni for the activation of lipoprotein lipase para ma-release ang mga fats nga naa sa chylomicrons and ang ApoE which is a ligand of the receptor of the liver para ma-process or ma-degrade sa chylomicron remnant inside of the liver. Okay? What about this one? Postprandial lipid metabolism. Postprandial means after meal. Especially if the meal is high in fats, morning mahitabo. Okay. This one is an adipocyte. So, this is a fat cell. And this one is chylomicron. Diba? Okay. Kanang mas klaro unta nga laser. Okay. Kani. Kani. Then, um, okay. Nganong na may glucose diri? Okay. Excess glucose and as well as excess of other monosaccharides. Um, they are converted by the fat cells have the ability not to convert this glucose into fatty acids. Okay? Yeah, especially if, di ba, general knowledge naman na kung sobra ang carbohydrates nga puno ng glycogen, then ang glucose it turn into fatty acid. So, mao ni ang process, di ba? So, this will be um, detailed no sa final term. Okay? But yeah, mao na usually ang pathway. Fructose, unsay mahitabo sa fructose? Ang fructose guys, i-convert na siya sa liver to glucose and then ayha dayon siya i-convert to fatty acid. So any other monosaccharide other than glucose has to be converted to glucose first before sila magamit sa body. Okay? And many studies have shown nga kaning fructose na to mao ning mas makatambok yun. Okay? Kay kaning fructose man good mas stimulate niya ang fat synthesis sa body. Maunang mga beverages that are high in high fructose corn syrup such as kanang mga Coke, maunang maunang dili sila maayo no because you know if sobra then sobra-sobra pud ang fructose then daghan pud ang ma-produce nga fat. Okay? Um Yeah. Step 2, lipoprotein lipase acts on tag in chylomicrons causing free fatty acids and MAG to enter the adipocyte. So remember ha, katong akong gidiscuss, di ba? Si lipoprotein lipase mo na yung function. So MAG means mono acylglycerol or monoglyceride. So mauning fatty acid nga na ay bilin nga nga glycerol nga isa. Isa nga glycerol then isa ka fatty acid. Whereas kaning DAGs, diacylglycerol o diglycerides, isa ka glycerol then na ay duha nga nabilin nga fatty acid. Na acid-free fatty acids mauning na completely digest ang fat molecule. So na ay na form nga tulog yun nga fatty acids and one glycerol. Okay? And remember, katong glycerol nga na form, i-donate to siya sa muscle naman to mahitabo fat cell na may capacity no to process glycerol kay pwede na niya gamiton para ma-esterify ang tulo ka fatty acids para makaproduce og triglyceride molecules. Okay? Lipoprotein lipase acts on VLDL, so FFA and MAG enter the cell. So later ma-realize pud ninyo unsay function sa VLDL and IDL. Okay? Ang iyahapod nga function is fat transport to extrahepatic tissues. So actually same ra og function si chylomicron VLDL and IDL, which is yes ma'am, which is to transport fats to the extrahepatic tissues. Okay? Mauna ang function nila, mauna ang similar sila. The pathways favor energy storage as stag. Okay? So, if well-fed ang tao, then most definitely ang metabolism is towards fat synthesis. Okay? So, insulin plays a role in that because aside from glucose utilization, insulin also stimulates lipogenesis by promoting entry of glucose into the cell and by inhibiting the hormone-sensitive lipase that hydrolyzes the stored tag to FFA and glycerol. Okay. So, yeah, mo ni siya usually ang mahitabo no? Like right after you eat, no? Especially um, one that has high fat and high carbohydrate. Okay. What about this one? This is called postprandial lipid metabolism in the liver. So, uh, so kaning glucose, iyahang i-convert ni siya no to again, pwede ni siya ma-convert into um, fatty acids through acetyl-CoA. Okay? So moagi na siya glycolysis and then start sa Krebs cycle. Pero dili niya kompleto ng Krebs cycle kay mo undergo na siya, mo divert na siya og lahi nga dalan padulong sa fatty acid production or fatty acid synthesis. Ang glycerol actually pwede na siya ma-synthesize from glucose because there is an intermediate in the middle of the glycolysis um, step 5. Okay? Pwede na siya pwede na siya mo U-turn into glycerol production. And remember, glycerol is used para ma-esterify niya ang tulo ka fatty acids para makaproduce og tag. Okay? So, as you can see here, ang carbohydrate, pwede gid siya i-turn into fat. Diba? So, this is how we convert carbohydrates to fats. Okay? So, kani sa liver na ni ha, kay hepatocyte man ni. Okay? So, what about amino acids? Amino acids can also be converted to fats in the liver. We can just deaminate or we can just remove the amino group of the amino acids and then ang mabilin nga carbon skeleton mao nang pwede gamiton to fatty acid synthesis. Okay, so much of this will be discussed later on sa final term nato. As well as those free fatty acids that are complex with albumin, pwede na i-free ni albumin, no? Para ang free fatty acids ma-take up sa liver, then makatabang pud sa triglyceride production. CR or sa man eh. This one enters the hepatocyte by endocytosis and are taken up by a lysosome. So CR is chylomicron remnant. So mao na gini nga i-take up siya ni liver mao nitong step 7 ganina sa exogenous. di ba? So nganong i-take up man niya ning chylomicron remnant para katong mga nabilin pwede niya i-process no into, for example, para makaproduce og cholesterol, para makaproduce og bile, kanang mga phospholipids nga nabilin sa chylomicrons kay pwede niya i-turn into phospholipids, di ba? Pwede po na niya gamiton para makaproduce og VLDL, which is the first step no of the endogenous lipid metabolism. Katong mga nabilin nga gamay nga fatty acid and glycerol, pwede po na niya i-convert to fat. Okay? So everything is recycled, di ba? Ang chylomicron nga gi-produce dito sa intestines para mo deliver atong mga fats nga gikan sa atong diet, kung nahatagan na og share ang muscles and adipocytes, then naa gyud na mabilin, and that's the remnant. And dili gyud na masayang ang remnant. Kaning leftover kinahanglan na siya i-utilize ni liver para makaproduce sa kaning mga substances. Okay? And this is now the start of the second type of lipoprotein transport, which is the endogenous lipid metabolism. So, this starts with VLDL production. Okay, so what na chylomicron diri? Ang first nga lipoprotein nga mo produce is VLDL. Okay? Yeah. And um diet can influence the production of VLDL through this, di ba? Okay. Kanang mga remnants sa chylomicron, pwede man na gamiton as starting materials para makaproduce og VLDL, which is now the start of the endogenous lipid transport. Okay, step 1, nascent VLDL are made in the Golgi apparatus of the liver. So sa Golgi apparatus sa hepatocytes no diha ma-produce si VLDL kay di ba si Golgi maunang iyahang dili ninyo ma-recall ang function ni Golgi apparatus, di ba siya ang tigpak sa mga molecules nga naa sa cell. So ang proteins nga na-produce sa rough endoplasmic reticulum, then ang fats nga na-produce sa smooth endoplasmic reticulum, i-process na ni Golgi apparatus. So, especially sa liver, ang function ni Golgi is kato mga na-produce nga proteins and lipids, i-turn niya into VLDL. Okay? Remember that it's the B100 no ang naa diri sa nascent VLDL, and naasad apo E. Okay? Sa step 2, additional apolipoprotein CNE are transferred from the HDL. So same ra gyapon sa exogenous nga si HDL mo donate gyapon siya og C2 and ApoE sa VLDL. And same ra gyapon didto nga ang purpose ni ApoC2 or ni ApoC is ma-activate ang lipoprotein lipase para ma-transport ang fat from VLDL to the extrahepatic tissues. Okay? Yeah. And as the VLDL shrinks in size, so ma-transform pud siya into something else. Like if mugamay siya, then mahimo siyang IDL later on. And then si IDL, if ma-exhaust pa gyud niya ang iyahang mga fats, pwede na siya ma-turn into LDL, which is now the smallest form of lipoprotein involved in the endogenous lipid metabolism. So, dili na mag-make sense ang ilahang differences sa components nila kay remember di ba si VLDL mas dako kay LDL. Ngano man na kay si VLDL later ma-turn man na siya into LDL after ma-release ni VLDL ang mga fats to the hepatic tissues. Okay? So, as the tag is removed from the VLDL, the particle becomes smaller and becomes an IDL. Diba? So, further loss of tag and it becomes an LDL. Diba? Oh, diri nga part no, gibalik ni di VLDL ang apo C kay HDL para si LDL sa si VLDL may mong IDL. As you can see here, si IDL wa na apo C. Okay? Oh. So, sa maning step 6, LDL are taken up by the LDL receptors found in the liver and non-hepatic tissues. So, um, katong dito sa si chylomicron remnant is taken up by the liver through the interaction of the chylomicron receptor and ApoE. But in this case, wala na nag-participate si ApoE. So, ang nag-participate na si ApoB100, which is now the ligand for this very famous receptor called LDL receptors. Okay? So, si LDL receptors, iyang function is i-take up si LDL from the plasma, internalize niya. So, kaon niya ang LDL from the blood to the inside. Okay? And para ma-internalize na, kinahanglan nga present si ApoB100 para ma-recognize siya ni LDL receptors para maka-undergo endocytosis. Okay? So, inana siya, no? So, maunang, diri pud mag-make sense nganong si LDL, gitawag siya nga bad cholesterol because iyahang i-donate pud ang cholesterol dili lang kay liver but also sa extrahepatic tissues like muscles and adipose tissues. So, um, para mas masabtan no, si VLDL ug IDL ilahang purpose is to give up the fats. But si LDL now, since mag-expect ta nga wa na kayo ni fat kay nawala na, maulagi na siya nga nahimo siya from VLDL and LDL kay wala na siya fats. So ang nabili niya is cholesterol na lang. So therefore, si LDL nimo ang final nga step niya no is i-internalize na siya ni liver and the other tissues para magamit ang cholesterol nga naa sa ilaha. Now, what happens if taas imuhang cholesterol intake or if taas ang imuhang intake sa saturated fats and trans fats which are deemed as bad fats, di ba? Bad type of fats. The reason relies um on the LDL receptor. Okay? Studies have shown that if imuhang diet is taas sa saturated fat and trans fat, mugamay ang production sa LDL receptor sa imong mga cells. So imagine if wala na yung LDL receptor nga ma-produce sa surface sa mga tissues, then LDL cholesterol would elevate in the blood. Okay? So, naara ni siya sa blood mo accumulate kay unsaon man pag-internalize kung walay LDL receptor. So mao na ang reason nganong unsa ni nganong bad ang saturated fats because if excessive yung saturated fat intake mo decrease ang production sa LDL receptor, so wa nay magpasulod kay LDL, so si LDL mo taas ra siya sa blood. And kanang mga cholesterol nga naa diha sa LDL, pwede na siya ma-oxidize and pwede na mag-build up sa arteries that can cause atherosclerosis and later on heart attack. Diba? So, I hope na realize ninyo nganong dili pwedeng excessive atong saturated fats and trans fats tungod ang na. Okay? Yeah. So, again, LDL delivers cholesterol to the tissues. Okay? So, it's very important for us to really study lipoproteins no in this level, in the molecular level para mas masabtan nato ang nganong inana, nganong mauni ang mga principles sa nutrition. Okay? So before wa na to kayo nasabtan nga nga saturated fats kay um ganong bad siya. Okay? But karon mas masabtan na nato tungod ani. Okay? Oh, so take note ha sa mga receptors, take note sa mga mga lipoproteins nga involved that, okay, sa mga apoproteins nga involved that. Okay? What about reverse cholesterol transport? So reverse cholesterol transport mao ni ikatulo, and this is being coordinated solely by the HDL. So iyahang function guys is iyahang kuhaon ang mga extra cholesterol from the tissues, from the extrahepatic tissues to the liver. Okay, mao ni iyahang function. So bali kay LDL, si LDL iyahang ihatod ang mga cholesterol from itself to the tissues, but si HDL iyahang kuhaon ang mga cholesterol from the tissues to the liver. Okay? So this is another route of the body nga ginahimo niya para ma unsa ni ma-excrete niya ang extra nga cholesterol for bile excretion. Okay? Kay ang end product man ani is bile. Then kaning bile i-excrete man ni sa body after niya ma-perform iyahang function sa fat digestion, then ang extra nga bile kay mupadulong biya sa tae or sa stool. So that is how we transport excess cholesterol through bile excretion, bile production and bile excretion. Okay. So first, unsa may mahitabo sa first step, lipid-free ApoA, or this is just the pure protein, pure apoprotein, is secreted by the liver and intestine. It is also released from chylomicrons and VLDL during TAG hydrolysis. Okay. So, ah, sorry guys, medyo saba. Wait lang. Okay. So, it starts with the production of ApoA. Okay. So, si ApoA again ma-release ni small intestines and the liver. So, next is ApoA acquires phospholipid and cholesterol from interaction with liver ABCA1, resulting in nascent HDL particles. Next dayon is mag-donate dayon si liver og phospholipid and cholesterol, and para maduna siya kinahanglan sa nga mag-interact si ABCA1, which is a receptor in the liver, with the ApoA. So the function of ApoA is to acquire phospholipid and cholesterol from the liver for assembly of HDL. So since nabutangan naman og phospholipid and cholesterol ang ApoA, so mahimo na siyang lipoprotein. Okay? But kaning end sa step 2, dili pa ni siya ang mature HDL, kay nascent pa ni. When say nascent, immature pa, or in this case discoidal. So how does nascent turn into mature HDL? They acquire more PL and cholesterol via ABCA1. Okay? So mudako gamay lang ang kuan. Just a little bit lang nga pagdako para ang nascent mahimong mature HDL, and para mahimo na mo-acquire siya og additional phospholipid and cholesterol from the non-hepatic tissues. So, para makaproduce og nascent, ang cholesterol dapat magikan sa liver. But para mag-mature ang nascent into mature HDL, kinahanglan nga ang HDL magkuha og additional phospholipids from non-hepatic tissues. Okay? So, diri nato ma-realize no ang function ng HDL, which is to cleanse the tissues of extra cholesterol. Kay actually mura siya kanang garbage truck nga muagi siya sa mga tissues just to collect the extra cholesterol. And it and HDL does that through this ABCA1 nga naa sa surface sa non-hepatic tissues. Then si ApoA, once nga mo bind siya with this ABCA1, then it allows the exit of cholesterol from the inside of the non-hepatic tissues. So, maunang mahitabo para ang nascent mahimong mature HDL. So, di ba ga-collect siya og cholesterol. So, yeah, diri na to ma-realize ang iyahang general function. Then, step 4, the enzyme LCAT or um, lecithin cholesterol acetyltransferase carried on HDL particles esterifies C to CE that migrate to the particle core. So, kinahanglan nga ma-esterify sa cholesterol para ma-internalize ang cholesterol. Kalima, the now spherical mature HDL continue to acquire PL and C via ABCG1 and C via SRB1 in peripheral tissues. So para mas mag-collect pagid siya og daghan nga cholesterol, kinahanglan nga kaning si HDL mo through its ApoA mo bind with SRB1 nga naa sa non-hepatic tissues para to allow no the additional exit of cholesterol. And LCAT continues to sterify C to C forming larger HDL. So as the cholesterol is being internalized, mudako pud ang HDL. So some CE are transferred to VLDL and LDL mediated by CETP or cholesterol ester transport protein. So some of the cholesterol esters are also transferred to VLDL and LDL mediated by this protein. And then the last step, the liver SRB1 binds HDL. CE may be selectively removed of the HDL particle, may be internalized and degraded. So through the binding with the SRB1, which is on the surface of the liver, it allows no the internalization or the degradation of the HDL into cholesterol. And sa diri nga part, diri masugdan no ang pag-excrete sa too much cholesterol. Kay kaning cholesterol, it turn siya into bile for excretion. Okay? So this is how the HDL cleanses the body of cholesterol. Okay? Through a series of, cholesterol sequestration from the tissues. Okay? So again, remember the enzymes, remember the proteins involved, the receptors involved. Okay? So that's it. Medyo taas-taas atong discussion but it's worth it since mas masabtan nato ang lipoprotein metabolism, and this is what really happens inside the body for lipid transport. Okay, so I hope mas nasabtan ninyo siya, and I hope mas kuan siya no, makasupport siya sa inyong um, para mas able ta no nga make og mga interventions to treat underlying nutritional disorders. So I guess I have to end this third part na ni siya sa atong discussion, kay mobalhin ko sa other room kay para dili kayo saba. Thank you.